Hello, every professors. I'm Zheng from Nankai University and Tianjin Third Central Hospital. It's my honor to give a presentation. My topic is about the PIK3R1, SBMB2, and CRYAB as potential biomarkers for patients with diabetes and developing acute myocardial infarction. Now I want to introduce from 5 parts. First, introduction. In the lesson 2017, there's a global composition of a number of deaths by level 2 caused by 23 GBD age groups, both sexes combined, and demonstrating that with age, the cardiovascular disease-related mortality and morbidity increased in the 3 decades. And the in-hospital mortality is about 20% about the myocardial injury, and the type 2 myocardial infarction mortality is 13%. And the long-term all-cause and cardiovascular mortality about the type 2 myocardial infarction is 1.3% as the major mortality in the cardiovascular disease. And in the right figure, demonstrate that the hypertension, arrhythmia, and heart failure is the major cause of the type 2 myocardial infarction associated mortality, and arrest sepsis is not the major but minor. And the patients with acute myocardial infarction and diabetes has less survival rate compared to the patients with non-acute myocardial infarction and diabetes. And in the right, use the treatment for the myocardial infarction and diabetes. The protective effect has risen. So we think about that. Can we get some biomarkers for diagnostic or prognostic for the patients with myocardial infarction and diabetes to increase the survival rate and decrease the mortality and morbidity? So we first use the 6 geo datasets, including GSE 775, GSE 19322, GSE 97494. The 3 is the acute myocardial infarction geo datasets, and GSE 3313, GSE 10617, and GSE 136948. The 3 is the diabetes geo dataset. After that, we identify the differential express genes, DG, and do the integrity bioinformatic analysis. Using Venn diagram, we obtain the same transcript between the diabetes and acute myocardial infarction datasets, and further GO and KJJ enrichment pathway analysis will carry out. And PBI, protein-protein interaction network, was done to obtain the hub genes, and finally the validation. We validate using the 2 ways. First, using mice model. Second, using human blood sample. Using mouse model, feed with high-fat diet for 3 months and administered with STZ. And the diabetic mice model was constructed. Then this mice will construct the myocardial infarction model, and the echocardiographic examination was also done. qPCR and ELISA of the left ventricle samples were done too, and the correlation analysis were also done. The second, we validate using human blood samples. We collect the human blood samples and to do the ELISA of the serum After that, we also collect the collection information and the laboratory examination results and do the correlation analysis between these parameters and the ELISA results. The human blood sample was collected twice, just a minute, and immediately discharged. The inclusive criteria and the exclusive criteria were as follows. And the third part is the results. The table 1 is about the 6 geodata sites information. And first, we do the integrity bioinformatic analysis of the acute myocardial infarction geodata sites. In the figure C, we found that the acute myocardial infarction and the control is separately, and it can be further analysis. The figure G is the protein-protein interaction network of the DGs using the acute myocardial infarction geodata sites. The same analysis was also done to the diabetes geodata sites. The figure C is the 3D PCA diagram, and the figure E is the DG protein-protein interaction network. And using Venn diagram, the same transcript between the acute myocardial infarction and the diabetes were obtained. 67 DGs were obtained. And table 3 is the DGs information, including upregulated and downregulated. And further GO-PGG enrichment pathway analysis were carried out. And in the figure F, the PPI were done to obtain the hub gene, including PIK3R1, SPNB2, and CRYAB. The 3 hub genes were validated. This is the microRNA hub gene network. In the figure D, we found that microRNA 153 3P can interact with the PIK3R1 and SPNB2, which may be the potential biomarkers for the treatment with the patients. And the validation part. First, we use the mice model to validate, and the figure A to C is the qPCR results. We found that the PIK3R1 and SPNB2 expression in MI group was higher than the control group, while the CRAB at 1 day was lower expressed in the MI group, and there's no significant at 1 week after myocardial infarction. And the figure D to F is the ELISA result of the left ventricular samples. And the PIK3R1 expression and SPNB2 expression is the same trends with the qPCR results. Interestingly, the CRYAB expression at 1 week is lowly expressed in the MI group. Well, this is significant differences at 1 week. And so maybe there's underlying post-translational modification as the interesting targets. And we also do the correlation analysis between the left ventricular ejection fraction and the hub gene expression, and demonstrating that the PIK3R1 and SPNB2 is negatively correlated to the ejection fraction, while the CRYAB is positively correlated to the ejection fraction. Next, we validate using human blood sample. The supplementary table 1 and table 2 is the parameters. The supplementary table 1 is the baseline between the acute myocardial infarction patients with diabetes and only diabetic patients, and demonstrate that the blood pressure, CK, CKMB, CTI, BNP, ALT, AST as significant differences. And table 2 is the parameters before and after treatment, demonstrating that the CK, CKMB, CTI, AST as the differences between the before and after treatment. And the figure A is the Hub-gene expression. We found that the PIK3R1 SBMB2 is highly expressed in the acute myocardial infarction group, and the CRYAB is lowly expressed in the acute myocardial infarction group. To investigate the diagnostic value of 3 Hub genes, we do the RC analysis. The 3 Hub gene can be the potential diagnostic biomarkers for the diabetic patients with acute myocardial infarction or without. The figure D to F is the expression before and after treatment. PIK3R1 and SPMB2 expression became low after treatment, and the CRYAB expression increased after treatment. And figure I also illustrates that the lowly CRYAB expression patients seems to have higher hospital stays. The figure G is to explore the prognostic value of the 3 Hub genes. We also found that the 3 Hub gene can be the potential prognostic values due to the AOCs. And we also do the correlation analysis between the parameters and the 3 Hub gene expressions. And we found that the CK, CKMB, CTNI, AST is negatively correlated to the PIK3R1 and SPMB2 expression, and negatively correlated to the CRYAB expression. And there's no significant differences between the BNP and the ALT and the 3 Hub genes. The part 4 discussion. The mortality and morbidity of patients with diabetes and undergoing acute myocardial infarction are still increasing, and the increase in hospital mortality and morbidity is mainly driven by the underlying cardiorenal dysfunction. So, the integrity bioinformatic analysis may be helpful and provide biomarkers for the patients. The PIK3R1 somatic variation can attribute to the genetic variation in the PIK-AKT pathway, and the PIK enzyme complex as a primary present in the immune system, and may be in part and dysregulated in some immunity disease, for instance B-cell lymphoma, and maybe influence some immune cells, such as the macrophage. And the second, SPMB2 is an essential component of the red blood cell membrane skeleton, and the downstream is the TGF-beta. SPMB2 deletion in mice contribute to the activation of TGF-beta SMAD signaling, and maybe can prevent properly development of a heart, and maybe is the potential biomarkers to treatment the diabetic patients with acute myocardial infarction. CRYAB can retain fundamental functions in straight muscle during the cytology and pathology modification. An inhibition of the UBE2V1 decreases CRYAB receptor 120G as a mutant CRYAB in cardiomyocytes and induced aggregate formation through enhanced proteasome system performance. And treatment these targets can treat and increase the heart failure, and which may be provide a novel therapy for cardiac disease with increased post-toxic stress. Also, there's some limitations. First, the HAP gene was validated using the mouse model and human blood sample. However, there are some other neighborhood genes, and due to the enrichment method and validation method, there may be some false negativity. So there's still needed to validate the neighborhood gene. It can also may be the potential biomarkers. Second, we aim to investigate the potential targets. So we only discuss a few significant genes, and maybe the others is also important. Lastly, although the sample size is not too large, but after evaluation, they still meet the requirement for further enrichment analysis and other statistical methods. Last, conclusion part. The screen hub gene PLK3R1, SBMB2, and CRYAB has been validated as a credible molecular biomarkers using the mice model and human blood samples, which may provide a novel therapy for the cardiac disease. Thank you.